Well, thank you, David. It's a very kind award or introduction, and I am really honored to be named as the House Scholar. It's definitely a privilege and an honor, and I am very excited to be part of the legacy of both Fred House and the others who have been named House Scholar before me. It's a truly excellent group to be a part of. So let me see, this is the screen that's got my stuff on it. All right, so I'm going to talk to you today about some work I've done in mathematical modeling for pharmaceutical applications, and it really does fit in well with the keynote speaker we had this morning, and I didn't know when I submitted my abstract for this talk that it would have such a nice overlap and have some nice synergy with our keynote. So in the current drug discovery pipeline, for every 100 drug candidate molecules, only one will ultimately make it through all of the clinical trials and be FDA approved for use in human patients. And including all of the attrition, this costs about $1 billion to develop one single new drug for clinical use. And this process takes on the order of 13 years typically for development of a single new drug from the candidate stages all the way to the end. And so there's vast room for improvement in this pipeline. And so some of the work that our keynote speaker talked about was addressing the discovery phase of the pipeline. And I'm more interested in things downstream of that. So I'll be talking a bit about what that means in a moment. I'm also very interested in things that shift the paradigm. So currently, you may take pills or syrups, typical dosage forms. You think about having a dose every day, or multiple kinds of medicines that you take every day or every 4 to 6 hours. And there are these kind of assistive aids, especially for the elderly or people who have lots of kinds of medicines to keep track of. And for a typical cancer patient, you may have 5 or 6 pills that you may need to take on different kinds of dosing schedules. You may need to take some with food, some later. So it may not even be one a day. It may be different periods throughout the day. And in this, there's like a morning, evening, afternoon kind of with this platform. But really trying to shift towards making things more patient-friendly where they comply with the dosing regimen, where they actually continue the entire regimen and don't drop off because they forget doses, they forget to go to the pharmacy, they're unable to take them at the proper schedule that's going to have the optimal therapeutic benefit. And so really trying to shift the paradigm and think more like having a dose once a month or longer term. And so this may not be a pill form, but maybe something more sophisticated. But then it's much easier and patient-friendly for the patients. So controlled release drug delivery is the main technology that I'm interested in. So traditional drug delivery systems are things like pills, capsules, injectables, syrups, all of the kinds of things you typically have in your local pharmacy, also topical creams, things like that. So with those, you have some time where you inject the dosage, or take the dosage, you may be digesting it, and then there's some peak in concentration, and then there'll be a decay in time fairly quickly after that as it's metabolized or eliminated from the body. And then if you want to have a long-term extended therapeutic effect, you need to repeat your doses. And these doses are generally empirically tuned so that you don't have peaks in toxic region concentration levels that would have very harmful side effects or toxic implications, but also try to minimize the valleys in the ineffective region where the medicine is not having any therapeutic effect. With controlled release technologies, you could have a single dose and have an extended period of time before there's a drop-off in the concentration of the drug. And so there are some applications that are already FDA approved. And so this is a cartoon schematic for a Lupron depot. So this delivers a hormone therapy for testicular cancer, and it's about the size of a matchstick. And it's implanted generally in the forearm of the patient. And the osmotic pressure from the biological fluids presses on this little piston, and it slowly releases drug molecules into the bloodstream. And so the patient has 1 to 3 months of constant hormone dosage. And so this is very beneficial. And then when this dose is finished, the implant has to be surgically removed, but it's a very local kind of surgery. I'm interested in this polylactic co-glycolic acid is a polymer, PLGA, and these actually completely biodegrade in human tissue, and they don't need to be removed afterwards, and their byproducts are biocompatible. That means that they don't have harmful side effects. They're already present in human tissues. So today I'm going to be talking about 3 main domains that I've worked in relative to pharmaceuticals. So first is formulation manufacturing. So once you have a drug compound that you want to deliver into a patient, you need to actually put it in something where they can take it, either to swallow it or a long-term release device. And so I represent it with this cartoon where there's something coating the drug molecules to get them into the body. Then there's also the issue of pharmacokinetics and pharmacodynamics. So once you put it in the body, where does it end up in the body? And what is its effect locally on the tissue where it's either administered or where its final destination is inside the body? And then drug release dynamics. So when you have the formulation, how does the drug molecule then get out of the formulation and act locally in the tissues? So first I'll talk about formulation manufacturing, and this has been the focus of my postdoc work at MIT. We work very closely with some pharmaceutical companies there with a large collaboration on campus. And so they have developed a process for continuous manufacturing. So these are very new kinds of manufacturing techniques in pharmaceutical industry. And so in this application, there's some solution of polymers and other materials that have the drug compound inside of them, and they are deposited on a plastic film, and it starts here as a liquid and it goes through this dryer, and this is basically a conveyor belt, and there's airstream and there's heated air underneath, and by the end of the process what you get out looks kind of like fruit by the foot or a Listerine oral dissolvable strip, so very thin edible film that, and in this case it would have some drug molecules dispersed throughout it. And so if you're going to be able to dose these to people, you need to have very precise control of the thickness of these materials and also the composition of, or the concentration of drug inside of it, and various other materials properties for downstream processing. You don't want it to be too moist, to be too sticky, or too dry where it's very brittle and it doesn't maintain the product integrity. And so a colleague and I worked on modeling the process here, and he worked on implementing process controls on the system. So for process controls in general, you have some inputs that you can actually interact with, some things you can change. So in our system, we could change the flow rate of the fluid onto the film, but we couldn't change the fan speed. We couldn't change how fast the air was moving on top. We could change the temperatures inside the dryer. And so there are some disturbance inputs, things we can't change, humidity in the room, for example, and they have some impact on the output that we care about and some outputs that maybe we can't measure but that are also still important. And so you use a controller, which is basically a set of sensors and a computer that manipulates the inputs that you can change and makes hopefully a positive impact on your output to maintain product quality. So if you don't know what's going on inside, it's more of a black box and you just tune the input to the output. But with a process model, which is what I worked on developing, you can actually map the output from the input. And so to do that, the model we developed involves simultaneous heat and mass transport moving in a moving domain moving coordinate system around this conveyor belt. And so we have a shrinking film. We have constant properties in the airstream. We have convection and evaporation between this interface. And we have like shrinking film. We have a temperature profile inside of this film. We also have Y is the conveyor belt speed. And we have a concentration, solvent concentration change in that conveyor belt dimension. And then the fluid can actually that's evaporating can diffuse through this vertical direction too. So we have kind of multiple dimensions of heat and mass transfer here. And so putting those things together, we developed a dynamic model for the system. And this is just showing if you had a constant air temperature throughout the dryer, what the dynamics of the solvent concentration that remains in that film looks like. And so it's nonlinear, and at each of these different colors, that's a new zone of the conveyor. So there was the first horizontal stage, there was a vertical stage, and then an upside down horizontal stage. So that's what those indicate. And so if we had a target where we wanted our product to have a solvent content of 0.05, it takes about 2,200 seconds for that to happen. And then with a constant temperature, it actually continues to dry the film. And that may not be advantageous if if the product could become brittle, if it goes below a certain moisture threshold, or if it's consuming more energy to continue to dry the film that way. So we want to have the optimal air temperature profile to actually reach this target set point. So applying some control strategies, the details of these are included in our paper. So these are non-constant temperature profiles. That's the important thing to note. Then with those two different control algorithms, we can reach the set point concentration level in just about 1,400 seconds. So in the grand scheme of things, that's not too long or too much difference, but it's on the order of this process that's it's quite good. And so it can actually maintain the concentration at the set point through the rest of the film, and other properties are still changing at that point too. So so this was very helpful for being able to better design and control this drying process. And so now I'll switch gears and move into pharmacokinetics and pharmacodynamics, and this is work that I'm just getting into. So pharmacokinetics, just a quick definition, involves the body's effect on the drug. So once you take the dosage and it gets into your circulation or whatever system it's administered to, where does it end up? So it may move between the circulatory system and a tissue, and it may go to the target tissue, or it may go somewhere else. It may also be eliminated through the liver or kidney or other kinds of excretion systems. And it also may interact with the lining of the intestine. And so this has huge implications with the target destination. So for example, with chemotherapeutic drugs, they're targeted towards fast dividing cells. And so when they're administered systemically throughout the whole body, some of the chemotherapeutic finds cancer cells and destroys them. Some finds cells that line the digestive tract that grow fast and attacks them. So you'll see cancer patients often are quite nauseous and just ill because they're facing these digestive issues. And also it targets nail and hair cells, which are also fast dividing. And so it has big impacts on non-targeted tissues that have similar properties. So that's pharmacokinetics. Pharmacodynamics is the flip of that, where it's the drug's effect on the body. And this can be systemic level effects, it could be organs, and all these multiple scales, so multiple length scales and multiple time scales of interactions. So tissue level, intracellular chemical signaling networks, and also just molecular kind of receptor interactions. And what we typically use to assess these kinds of things are external measurements of clinical kinds of indicators. And so to actually understand these effects from a clinical perspective is a big challenge. So I mentioned briefly about targets. So if you had some drug molecule that's been designed to have a specific kind of molecular interaction, maybe this is a receptor on a cell or some kind of intermediate inside the cell that needs to interact. Typically, they design these things to either have a charge interaction or some sort of fitting where it might be like a hand inside of a glove, where they're supposed to fit very carefully based on a molecular geometry. And there may be other things that have similar features other kinds of tissues, and it may interact with the drugs in ways that you maybe foresaw, maybe you didn't foresee. And also you could have other kinds of undesired targets where you didn't predict that it would have interactions, adverse side effects. And so these are interesting things that maybe could be explored more with computational methods before being used in the clinic. So there's a coupling between pharmacokinetics and pharmacodynamics, the drug's effect on the body and the body's effect on the drug. And so when you take a dose, you have to ingest it, or it has to get into your bloodstream somehow. The absorption, distribution, elimination, metabolism, all of these kinds of bodily functions have interactions with the drug. And then the drug may, at the molecular level or tissue level, have the effects that it's supposed to have, or things that are negative side effects or unexpected. So commonly, pharmacokinetics has been modeled by a series of reaction kinetics, and maybe moving them into compartments where the compartments, these may be not physiological kinds of compartments, but a central compartment where things happen fast and a peripheral compartment where things happen more slowly, and then some rate equations to connect these different regions. Recently, there's been more of a move towards physiologically based pharmacokinetics and pharmacodynamics, where it's actually considering compartments that are representing tissues and local tissues, and then bigger organ systems, and the exchanges of material between these different domains. And so you can have different kinds of interactions there. But generally, each of these compartments is assumed to be well-mixed reactors, and the compartments themselves are representing the spatial distribution of material. Traditional pharmacodynamics in pharmacology has been very empirical, where there's a clinically observed effect on the body that's recorded based on some drug concentration that's generally just a bolus injected directly into the bloodstream. And so there may be measurements of some desired effect and then some side effects. And so this is very empirical. And so we're trying to think about things more mathematically and more mechanistically. So others have gone the opposite direction and thought very mechanistically about what's going on inside the cell. And this is just showing there's a lot of details inside a cell with these reaction networks become quite complex with signals sent between different molecules inside the cell. And so systems biology, which is typically at the cellular level or subcellular level, uses a lot of information and a lot of these kind of graph theory type approaches. Systems pharmacology aims to kind of bridge these two regimes from the microscale molecular level events and the complex kind of clinical effects. So traditionally you had some primary, you had direct effects were thought to be as a consequence of interactions with a primary target, and then side effects with some off target. But in actuality, there's probably some complicated interplay between those things with the network. And so, so I'm very interested in actually considering the spatial effects of these kind of materials. And so this is looking this is more of a tissue level kind of approach, but looking at the transport of materials in a spatial domain. And so let me just explain this cartoon. So in the blue spheres are representing some cells. So in this paper, we considered them to be stem cells, and they needed some nutrients in order to divide to form a particular kind of tissue of interest. But these also could be tumor cells that need some medication in order to be destroyed. It could be all kinds of different cells. And these red spheres are representing some kind of drug delivery device located nearby that can deliver a targeted dose as a function of time. And so we formulated this problem as a one-dimensional reaction diffusion equation with convection. And so this PDE is describing the uptake or the movement of the material of interest, the concentration of the drug through this domain. So there's a convection term, there's a diffusion term, and this reaction term is representing the uptake rate of the nutrients into those cells. And we want the cells to actually have the desired uptake rate, so they have enough nutrients as a function of time for their proper cell metabolism. And so there's a minimization problem here where we want the actual, we want to minimize the difference between the desired and the actual uptake rates. And so this is my colleague took this as her PhD project and formulated as a theoretical process controls problem. And so if you're interested in the details of that, I refer you to this paper from last year, but also her dissertation has a lot of good control theory work. But basically, the novelty here was that using a spatial PDE, and where the constraint on the minimization problem was actually the boundary condition for a PDE. So it made this a very complicated problem. So now I'm going to switch gears again and focus on the rest of the talk on basically what I did as my PhD work in drug release dynamics. So I mentioned before that I'm very interested in biodegradable materials that you don't have to remove after they have given the patient the dose that they're designed to deliver. And so we use polymers for this purpose. And there are different kinds of mechanisms. That polymers are a very broad class of chemicals that are generally composed of repeating links of a chemical moiety that's just repeated. So it's kind of like the chains or the links in a chain. And so different kinds of polymers have different kinds of mechanisms. I'm very interested in those that are erosion controlled. And so this cartoon is just showing a sphere that has drug molecules throughout the interior. So these molecules would not be to scale for the size of the microsphere. And there's two different broad categories of materials that are erosion controlled. And so some are surface eroding. That means that they degrade by a reaction that has a reaction front where it degrades from the surface inward. And so over time, you have basically it looks like it's dissolving, kind of like a jawbreaker, something like that. And the drug is released as the particle is being dissolved or biodegraded. A more interesting or more complex type of material is bulk eroding. That means that the time frame for the media to get inside of this microsphere is very fast compared to reaction time. So in the order of minutes, this thing is completely hydrated with water, and then it takes months for the biodegradation to happen. And so slowly, pores form in this material as it reacts, and then the drug actually can move more easily through the pores that are formed if it's a large kind of protein-based drug. And so my work is focused on mechanistic models for understanding these kind of materials, and I've primarily focused on deterministic type models. So the material that I use most frequently is called polylactic coglycolic acid, and this is very ubiquitous in biomaterials. So they use it a lot for tissue engineering scaffolds, also for surgical sutures. It's FDA approved, and it's used in a lot of applications. And so it's a really exciting biomaterial. This polymer has two subunits, and these are not one is lactic acid, one is glycolic acid, and these are repeated, but they're not always all the X's and then all the Y's. They're randomly mixed together. And so these are the chains, and so it looks kind of like little spaghetti noodles throughout the inside of this, but it forms a solid. And it has tiny drug molecules throughout it, and water breaks this bond between the X and the Y group here, right? This ester bond. And so it forms two smaller chains, but those two smaller chains are both composed of this same polymer. And so the drug is released from this material, so it's kind of like a big solid ball of spaghetti noodles with the drug molecules in it. And eventually, as it reacts, it breaks some of these bonds, and there's bigger gaps for the drug molecules to diffuse out. And eventually, you have all of the drug is released, and all of the polymer is completely broken down. And when the polymer has very small fragments, those actually dissolve in water. So that is what forms the pores in these particles. So if this was a diffusion-controlled system, if you think about diffusion, if you have a constant diffusion coefficient, and these are spheres, and if a small microsphere would release its payload faster than a large microsphere with a constant diffusivity. That's what this plot is just showing. So the blue one on top is the smallest microsphere, the green one on the bottom is the largest. So this is just the standard diffusion equation. But what they noticed experimentally is that at some point it transitions from being diffusion controlled to something else. And so this was really the challenge, kind of the mystery that motivated my PhD work. How can we understand the mechanism that makes this happen? And so this is clearly not diffusion controlled, and it actually switches where the smallest microsphere, which is the blue one, releases its payload after the large one completely does. So there's something else going on. And so we have other experimental evidence that suggests things that may be part of this mechanism. So this is showing pH inside the microsphere. So these are based on size. So this is a 14 micron diameter, and this is 38. And so you can see that there's a larger acid concentration in the center of the large microspheres, but it kind of starts out just in the center and then it spreads. And so there's something with acid accumulation. And this polymer, the carboxylic acid in group, is acidic, is a stronger acid than the hydroxyl group, and it can't, this reaction, breaking this apart, is catalyzed by acid. So we believe it's being self-catalyzed by its own end group. Every time it breaks apart, there's a new chain that also has an acid on it. Also, this is some SEM images of the small microspheres versus large, and you can see that there's much more significant erosion of the morphology of these microspheres. So on the surface and in the interior, these have very hollow regions, and this is just slightly porous. So there's a difference in the large and small microspheres in regards to the pore formation. So my model that I've developed includes acid accumulation and pore growth dynamics in these small and large microspheres. And with the acid accumulation, I mentioned that the water hydrates these microspheres very quickly. So initially, their rate of breaking down, biodegrading, is very similar. After some time, these arrows are representing short diffusion lengths. So in a small microsphere, everything is a short diffusion length. Everything that's produced can diffuse out easily. With the larger ones, the surface, there's a much shorter or much limited range of places where the diffusion is very quick. And so in the center, this is diffusion limited, so the acids are accumulating there and are self-catalyzing, and they continue to catalyze the reaction where they form this hollow interior. So that's what's included in the mechanism for the model. And in the pore, you have a drug molecule, if it's smaller or comparable, or if this opening is too small, the drug molecule can't diffuse through it. But as the chain with this acidic end group, you form two chains, both with the acidic end group. If this is sufficiently small, it will dissolve and open up the pore. It will make it larger. And so then the drug can move through that more easily. And this is just showing the time scales involved in the process. So you have some hydration. I told you that was on the order of minutes, when this reaction can occur on the order of months. And so then for the bulk of the time, we have this reaction with water, aqueous fluid happening, and the pores are growing, and the drug can has increasing mobility through those pores. And then at the end, there's some rapid mass loss. So most of the model is focused on this time domain. And so there's many kernels involved in this model, and I'm not going to pretend to do my entire PhD dissertation defense in this talk after I've already done some other introductory things. So I'm going to focus on things dealing with the pore growth rate. So what happens with the polymer itself, not necessarily with the drug. And that has two components, acid and its accumulation and diffusion, and the polymer reaction. So I have a reaction diffusion equation in spherical coordinates. So it has the diffusion of the acid, acidic end group, and the reaction with the polymer. And so I have three species that I'm considering here. The polymer chains, which are P sub N, and I'll explain why that's P sub N in the next slide. The ester bonds, which are all of these linkages between the repeating units, so the links between the chains. And then the acid, each of these polymer chains has one acidic end group. And I've got them color-coded through the rest of this, so it's a little easier to distinguish between things. So the polymer doesn't exist as just polymer. It has a distribution of chains that are various lengths. And so the N represents the length of the chain. So a monomer has 1 repeating unit. A dimer has 2 repeating units. And that goes on to whatever number. And in this case, based on these molecular weight distributions, the largest polymer chain has approximately 10,000 repeating chains in it. And so to characterize the system, you would need to know all of those, the composition of all of these p sub n's from 1 to 10,000. And they react. Each polymer chain of length n can be formed by chains larger than it breaking, and it can then also break to form all of the chains smaller than it. So there's a reaction, a generation and consumption term there. So I already mentioned that each of the polymer chains has 1 end group. And if there are n of these x's and y's, then there's n minus 1 of the bonds between them. So that's what the ester is representing there. So the naive way to solve this would be to have the ester, the acid, and all 10,000 of the polymer chains to fully characterize the system. And you have all the information you need. You have the reaction, that's the proper order. You have the diffusion. But this is very expensive. And so as a second-year graduate student trying to run this on MATLAB, it actually ran out of memory on my computer. And so I'm like, well, that's not the way to go. And these nonlinear interaction terms make it where it would be difficult to run it in parallel because they all interact at every time step, and then the time steps are sequential. And so I needed to think of a way to do this as an alternative. So that was the full distribution method. The analytical reaction method is what's commonly used in the literature. And so basically they only treat the acid, and they only treat a first order reaction. So it's not interacting with anything else, and they completely eliminate all the diffusion. So basically this is saying that in every size microsphere, no matter how big or how small, all the reactions happening exactly the same, and there's no transport effects of the acid being accumulated, which does not agree with the experimental evidence I showed with the pH being different in the centers of these spheres. So this is very common. It's very easy because this is a first order differential equation that you solve in undergraduate DiffyQ and 1 equation, but you lose all the diffusion information. And so this is what's standard in this domain to consider this kind of reaction without the diffusion effects. And so I developed a method that still considers only the acid, but has accounted for the diffusion of the acid by saying even though only the small ones, the small polymer chains can dissolve, having some average diffusivity over all the chains where this can be solved analytically. And so with that information, you have some feel for what are the critical parameters that influence the transition between being diffusion controlled and being reaction controlled. And you have the simplified reaction term that doesn't depend on other things, but it's an improvement over the first order assumption and without diffusion that is common. So this is the analytical reaction of diffusion. The most common method for including the polymer but not including diffusion that's used that has a second order dependence on the reaction where the acid actually depends on interactions with the ester. This still treats that the acid is actually attached to the polymer. And so it only considers the reaction kinetics. This is the method of moments. Oh. Yeah. So the method of moments, again, loses this diffusion information that's very rich in telling you what happens in different sizes of microspheres. And so this is a very common traditional technique for population balances, and you get information about how many total end groups there are, which is the acid part, and how many total bonds there are, which is this ester part. But it's missing that diffusion information. So I started to think about the fact that I needed a method that was in between that full distribution and the very simple methods, but could capture this diffusion effect. So I started to think about, okay, maybe I could bin and lump some of these sizes of polymer chains together and move from 10,000 equations to maybe 100, something like that. But then through this analysis, it became clear to me, evident, that the diffusion, I didn't need the diffusion information for any of the large polymer chains because they weren't moving anywhere. They react, but they're stationary. And so, you only need to have the diffusion of the small polymer chains inside the microspheres. And so this method it has explicit consideration of the small ones, which is from monomers up to polymers that have 9 chains on them, or 9 linkages connected. And then it has the acid and the ester and the sum of the large ones. So it preserves it makes the non-mobile chains one big lump together because they're only needed to find out how many bigger chains can react to form small ones. And so through some algebraic manipulations, I can find that information without having to solve a unique PDE for that equation. And so that method, now I can solve the full system in about 10 minutes on my PC rather than running out of memory in MATLAB. So that's a vast improvement, and that's very exciting for me. So there's 2 methods that I've worked on. One is very simple but analytical, and so it gives you some information quickly. And then this other one involves a more sophisticated computational routine, but it does still, and it gives you the rich information on reaction and diffusion. So I'll show you a few results. These are results with just the analytical technique, the analytical reaction and diffusion. And so this parameter phi is a function of the reaction rate and the size of the microsphere divided by its diffusion coefficient. And so with a small microsphere here, this is in the diffusion controlled regime. As you move to larger microspheres, you start to see a peak in the acid concentration in the center that's very focused and centralized. And then eventually it spreads out and the acid concentration broadens, but it's still higher. And so this is very consistent with the experimental results. And also with that kind of model, we can understand the pore growth. And so these, the red planes are indicating a small drug molecule when the pores have exceeded the threshold of the size of a small molecule. And then the blue ones are a much larger drug molecule of protein, and it's showing when those, when the pores are large enough to release those particular drug compounds. And so the current state of the art was basically this, where there was no difference based on the size of the microsphere, where everything was uniform inside the microsphere domain. With the large, having this lumped parameter, the large polymer chains, the more sophisticated model, we can actually capture the transition from erosion or from diffusion controlled to erosion controlled, where we're capturing size-dependent autocatalytic PLGA degradation. So this was very exciting, and these curves are very sensitive to all the different parameters in the system, or when this transition occurs. So I'm still doing parameter exploration on here to understand fully under different conditions when this transition occurs and how severe it is. Here it's fairly late, where almost all of the small microsphere had released its payload by that time, but this also can transition much earlier, like the experimental data I showed earlier. So still working on that. So I, of course, want to acknowledge all of the funding sources and Krell and the DOE. It's been a fabulous experience. I wanted to show, this is the acknowledgement slide I showed when I finished the program. And really, such an important part of the program to me was the opportunities that I had as a fellow in this program. So opportunities to travel, opportunities to meet fascinating people. I was there when New York Blue started up at Brookhaven National Lab. That was very exciting. Opportunities to present my work and go sightseeing. This is John Zuhones in the back, and another Michael Bybee, who was there, also Illinois alumni who were part of this program. And so it's just very fun to meet the colleagues and interact with all of you. And so I'm so thankful for the opportunities that I have, that I'm very passionate about trying to help other people to have the same kinds of opportunities. And so I do a lot of hands-on stuff. Actually, Josh took a class with me in Illinois where we did hands-on stuff at the Boys and Girls Club, and we worked on science experiments. Many of these four are Girl Scout experiences that I've led in my hometown or in Oklahoma, in the area that I grew up in. And this is a group at MIT, actually all in my cubicle. This is the lab tour. We went into my cubicle, and I told them about basically this presentation of the work that I do. And so high school women getting excited about becoming scientists and engineers. And so these are all with girls, but I do other things. I'm not exclusive with girls. I do outreach stuff with boys too. The Boys and Girls Club was mixed. And so I just want to encourage you all to consider ways that you can be encouraging other people to have the opportunities, whether it's through outreach with children or just public advocacy for computational science and engineering or your specific scientific domain. It's very important. And also with your legislators, and that's important now. So with that, here's my overview slide, just summarizing the work that I've talked about, and I'll be happy to entertain any of your questions.